Have you ever wished you could simply delete a moment from your past? Perhaps you have experienced a painful heartbreak, a deeply embarrassing mistake, or a traumatic event that plays on a loop in your mind when you are trying to sleep. The idea of erasing a memory is one of the most persistent fantasies in human history. It appears in our movies, our ancient myths, and our secret hopes. We imagine a magical procedure or a pill that could act like a broom, sweeping away the mental cobwebs that make us sad, anxious, or afraid. But what is actually happening inside our heads when we form a memory? And is it physically possible to reverse that process and make a recollection vanish completely? To understand if memories can be erased, we first have to discard the idea that our brains function like video cameras. When you experience an event, your brain does not record a perfect, high-definition movie file that is stored in a single folder. Instead, an experience is broken down into pieces. The sound of a voice might be stored in one region, the visual of a face in another, and the feeling of fear in yet another. When you remember something, your brain is not hitting the play button on a video. It is actually rebuilding that moment from scratch, pulling these scattered pieces back together like a puzzle. Every single time you recall a memory, you are reconstructing it, and this process is surprisingly fragile. This fragility is the key to everything. Because a memory has to be rebuilt every time we think about it, there is a window of opportunity where the structure of that memory becomes unstable. Scientists call this process reconsolidation. Imagine building a sandcastle. Once it is dry and hard, it seems permanent. But if you pour water on it, the sand softens and becomes malleable again, allowing you to reshape it before it dries. Our memories work in a similar way. When you activate a memory, it briefly turns back into soft sand, and during that short period, it can be altered, strengthened, or weakened. This discovery shattered the old belief that long-term memories are permanent chemical scars in our brain. The very act of remembering makes a memory vulnerable, opening the door to the possibility of creating a true clean slate, if only we can find the right tools to gently sweep the sand away while it is still wet. To grasp how we might erase a memory, we need to look at what a memory is made of in the physical sense. It is not made of electrical energy that simply vanishes. A memory is a physical pathway in the brain, a connection between neurons, which are the nerve cells that make up our thinking organ. When you learn something new, your brain physically changes. It builds a bridge. This process is called synaptic plasticity. Imagine a dense, overgrown forest. The first time you walk through it to get to a destination, it is difficult and slow. You have to push branches aside and step over roots. But if you walk that same path every day, you eventually wear down the dirt, creating a clear, easy-to-follow trail. A memory is that trail. At the end of each neuron is a tiny gap called a synapse. To send a thought across this gap, the first neuron releases chemical messengers. The more you use a specific pathway, the stronger that connection becomes. This is often summarized by a simple rule: neurons that fire together, wire together. If you practice a piano scale or relive a traumatic moment, the neurons involved in that sequence send signals repeatedly. The sending neuron becomes more efficient at releasing its chemicals, and the receiving neuron builds more receptors to catch them. It is a physical construction project. Proteins are synthesized, and structures inside the cells grow, solidifying the path from one cell to the next. A long-term memory, therefore, is not a ghost. It is a physical structure of protein bridges that wasn't there before the event happened. This brings us to the central challenge of erasing a memory. To truly delete a memory, you cannot just empty the trash. You would have to physically break down those specific protein structures and prune away the synaptic connections that form the trail. This is a destructive process at a microscopic level. But here lies the danger. How do you destroy the bridge that represents the memory of a horrible car accident without accidentally burning down the bridges that represent how to drive a car or what your name is? The brain does not label these protein bridges with convenient tags. A single neuron can hold pieces of completely different memories. Trying to selectively dissolve the structural proteins of a single memory without wrecking the wiring of others remains one of the most complex biological engineering problems imaginable. The brain's wiring is a tangled web, and we are only just beginning to understand which threads are safe to cut and which would unravel our entire identity. For decades, scientists have searched for a chemical solution, a kind of mind-erasing drug that would remove the need for complex talk therapy. The goal is to target the reconsolidation window we talked about earlier. If a memory becomes unstable while being recalled, can we inject a chemical that stops it from hardening again, effectively washing away the sandcastle? This research has led to some fascinating and ethically complicated experiments. One of the most well-known drugs in this field is a common blood pressure medication called propranolol. This drug does not erase the factual details of an event. You will still know that the event happened. However, it blocks the action of adrenaline. Here's how it works. Emotional memories, particularly traumatic ones, have a powerful physical component. When you remember a terrifying event, your body reacts. Your heart races, your hands sweat, and a wave of fear washes over you. Propranolol is a beta blocker that prevents these physical reactions. The theory is profound. If you ask a person to recall a traumatic memory while under the influence of propranolol, the memory is reactivated and made soft, just like that wet sand. But because the drug blocks the fear response, the brain rewires the memory without the emotional punch. The memory is saved again, but this time, the bridge connecting the event to the visceral feeling of terror is gone. The memory becomes a narrative, a story of a bad thing that happened, rather than a horror movie that plays with the full orchestra of a racing heart and panic. However, this is not a clean erase. It is more like turning a high-definition color film into a faded, silent, black and white photograph. The skeleton of the memory remains. Other researchers have looked further upstream in the biological process, targeting protein synthesis directly. Just as your muscles need protein to grow, your memories need proteins to stabilize. Scientists can inject a chemical that blocks the creation of these proteins right when a memory is being recalled. In laboratory studies with animals, this has produced startling results. An animal trained to fear a sound can have that fear vanish completely if the protein blocker is present at the exact moment of recall. It is as if the trail in the forest disappears entirely. But these specific protein synthesis inhibitors are generally toxic to humans, and the idea of injecting them into a living human brain remains a distant and dangerous frontier, strictly limited to the laboratory for now. The search for precision brought us to a revolutionary technology that sounds like pure science fiction. It is called optogenetics, a word that combines light and genetics. The problem with chemicals is that they are messy. A pill travels through your bloodstream and washes over the entire brain, affecting millions of cells indiscriminately. But what if you could control specific neurons, the exact single pathway that holds a memory, with nothing but a pulse of light? Optogenetics allows scientists to do this, but currently only in laboratory animals, primarily mice. The process is incredibly intricate and reveals the exact address of a memory. First, scientists have to hijack a virus. Viruses are experts at entering cells and delivering genetic instructions. Researchers strip out the harmful parts of a virus and insert a gene from algae that produces a light-sensitive protein. They inject this harmless virus into a tiny specific region of a mouse's brain. The virus delivers the algae gene to the neurons. Now, only the neurons that are firing actively during a specific experience will be tagged. Suppose you give a mouse a mild shock in a specific chamber. The neurons that encode the memory of that fear are firing wildly. The genetic trick ensures that only these active neurons produce the light-sensitive protein. The memory cells have now become essentially light switches. Then, a fine fiber optic cable is implanted into the brain, aiming right at these tagged cells. When blue light is pulsed through the cable, it activates those specific fear neurons instantly. In landmark experiments, scientists implanted this technology and created a false memory. A mouse in a completely safe blue box had its fear memory neurons, which were formed in a different room, stimulated by light. The mouse instantly froze in fear, even though nothing bad had ever happened to it in the blue box. The mouse was remembering a shock it was not currently receiving. This proved that targeting a specific cellular ensemble was enough to hijack a memory. If we can activate a memory with light, the question becomes, can we erase it with light? By pulsing a different color of light to shut these neurons off, researchers have been able to turn off a learned fear, effectively providing a switch for the memory. While this is not permanent erasure of the wiring, it demonstrates a functional off switch, proving that the physical traces of memory can be directly manipulated in ways we never thought possible. If we want to erase memories, we should not just focus on waking science fiction techniques. We need to pay attention to what our brains do naturally every single night. Sleep is not simply a time when the body rests and the mind shuts off. Sleep is the time when your brain does its housekeeping, and a large part of that chore involves managing your memories. During the deep stages of sleep, your brain enters a process of synaptic homeostasis. Think of your brain during the day as a massive, messy desk. Every experience adds new papers to the pile. If you keep piling them up, the desk becomes chaotic and you cannot find anything. Sleep is the time when the janitor comes in. This janitorial process involves a literal weakening of connections. Your brain is not keeping every single thing you saw and felt during the day. It is selectively preserving the important information and actively discarding the noise. You do not remember the color of the fifth car you passed on your way to work, and you do not need to. While you sleep, your brain weakens the synaptic connections that represent the trivial, the mundane, and the useless clutter. This is a form of natural, nightly memory erasure that is essential for sanity. If we could not prune our mental trees, our brains would saturate, and we would be unable to learn anything new. The brain decides that the intense conversation you had is a keeper and strengthens that pathway, while letting the memory of the random background music dissolve into static. This has led to a new frontier in trauma therapy. Researchers are exploring whether we can manipulate sleep to actively erase targeted memories. The technique involves a principle called targeted memory reactivation. While a person is undergoing therapy for a phobia, a specific scent, perhaps a rose or lemon smell, is introduced so that the smell becomes associated with the therapeutic process of unlearning the fear. Then, while the person is sleeping deeply, that exact same scent is puffed into the air around them. The sleeping brain recognizes the scent. Because the brain is currently in maintenance mode, it does not just passively smell a rose. It reactivates the therapeutic memory associated with the smell and, crucially, reprocesses it. Studies have shown that people exposed to the scent during sleep have a dramatically reduced fear response the next day. The brain, acting as its own surgeon, used the scent as a trigger to weaken the fear pathway during the night. It is a gentle, non-invasive method of guiding the brain's natural delete function to erase the emotional sting of a bad memory while we are completely unconscious. While we wait for futuristic drugs and light technologies, we already possess a tool that can rewrite our past, and it is entirely natural. It is the process of talking. Psychotherapy, particularly methods designed for trauma, is essentially a biological intervention in the brain's memory system, and it works through the reconsolidation window we discussed earlier. The goal of therapy is not usually to delete the factual narrative. The goal is to disconnect the memory from the overwhelming physical panic that ruins a person's quality of life. This is not just wishful thinking or hand-holding. When done correctly, it physically changes the structure of the memory pathway. For a memory to be reconsolidated and updated, it must first be fully reactivated. In a safe therapeutic environment, a person is guided to bring the traumatic memory to the front of their mind. By describing the event in vivid detail, the brain opens the file and makes it malleable. The memory sits there like the wet sandcastle. But this time, the context is different. The person is sitting in a safe, comfortable room, speaking to a calm, empathetic professional. The brain is now facing a massive contradiction, a prediction error. The memory says, "This event means extreme danger and I will die." But the current reality says, "I am safe here in this chair." The brain cannot hold these two completely opposite realities at once. This conflict forces the brain to update the old memory file. With repeated sessions, the brain literally rewrites the emotional soundtrack of the memory. The pathway that linked the event to the amygdala, the brain's fear center, begins to shrink. A new pathway, one that links the event to a sense of context or logical understanding in the prefrontal cortex, grows stronger. The memory is not erased, but it is transformed. It moves from being a crippling, present-tense flashback to a past-tense narrative. Eye movement desensitization and reprocessing, or EMDR therapy, is a fascinating example of this. By recalling a trauma while moving the eyes back and forth, the patient is taxing their working memory. This makes the image of the trauma less vivid, as if the brain is pulling the picture so hard that it blurs. It is a physical workout for the brain that degrades the sharp edges of the nightmare, proving that targeted, non-chemical interventions can indeed reach into the brain and scrub the vividness away, leaving a duller, less painful trace behind. When we talk about erasing memories, we often confuse two very different processes: actual erasure and something called extinction. This distinction is crucial for understanding why old fears can come roaring back, even after we think we have conquered them. Suppose you have a fear of dogs because a dog bit you when you were a child. The original memory pairs the image of a dog with a sensation of pain and terror. To heal, you might go through exposure therapy. You slowly and safely interact with friendly dogs. Over time, you stop panicking when you see a dog. It feels like you have erased the fear memory. But is that truly what happened? Neuroscience tells us that the original fear memory is still there, sleeping. What you have done is build a brand new, second memory that sits on top of the old one. This new memory says, dogs are safe. The old one is a pathway that screams, dogs are dangerous. This new learning is called an extinction memory. The problem is that the brain has two tracks running side by side, and the old, dusty track of fear never really goes away. It is just suppressed by the strength of the new track. Under normal circumstances, the rational cortex keeps the fear pathway silenced. It is an inhibitory control, but the old pathway is still made of proteins. It is still physically wired in your brain. This is why relapse happens. If you are put under immense stress, if you are exhausted, or if you encounter a dog that looks exactly like the one that bit you in the exact same context, the old pathway can suddenly break through the inhibition. The fear comes flooding back as if you had never undergone therapy at all. Real erasure would mean actively tearing up the old train tracks, not just building a bypass road over them. The research into reconsolidation updating aims for a true rewrite. The goal is to open the original file and permanently alter it so that the fear response is not inhibited, but gone. This requires the brain to enter that specific fragile state where the memory can be directly modified before it is saved again. However, most of our daily forgetting, and even much of our therapy, is based on the less permanent process of extinction. We are not truly deleting our past. We are simply learning to tune it out. Understanding this paradox of extinction explains why treating deep-rooted trauma is so difficult and why a true erasure cure would have to chemically or optogenetically dismantle the old path, rather than just paving over it. If we do achieve the ability to perfectly erase a memory, we face a final, perhaps even greater, obstacle than the biology itself. That obstacle is the question of who we are without our scars. This moves the discussion from a scientific "can we" to a deeply philosophical "should we." Imagine a world where a simple medical procedure can delete the memory of a sexual assault, an act of betrayal, or a war zone. On the surface, this seems like the greatest gift medicine could give to a suffering mind. The pain would be gone, the nightmares would cease, and a person could finally live a life unburdened by the weight of horror. It sounds like pure mercy, but our memories are not isolated photographs in an album. They are the foundation stones of our character. If you erase a traumatic memory, you are not just deleting a bad feeling. You are potentially deleting the event that taught you empathy, the lesson that made you cautious in dangerous situations, or the grief that shaped how deeply you love. When we erase a memory, do we inadvertently erase a piece of our moral development? If we erase the guilt of a harmful action we committed, do we become more likely to repeat that action because the emotional consequence has been chemically scrubbed away? Our identity is a narrative, a story we tell ourselves about where we have been and who we have become. If we tear out the darkest chapters of our personal book, the plot may no longer make any sense. We might look in the mirror and see a stranger, a happy person who lacks the depth that suffering and recovery can bring. Furthermore, there is the profound issue of justice and truth. If a person is a witness to a crime, but they choose to erase the memory of that crime to ease their own suffering, what happens to the truth? Does the perpetrator go free because the evidence has been biologically deleted from the only brain that held it? These technologies risk turning memory from a sacred, if flawed, record of our existence into a commodity that can be shaped, sold, or erased at will. The human experience is not defined solely by moments of joy. It is defined by the entire spectrum, the integration of light and dark. To erase a memory is not to heal a wound. It may simply be to create a hole. And a person who is full of holes, with empty spaces where vital, painful lessons used to be, might not be a whole person at all. The ultimate goal might not be to erase the memory, but to transform it so that it loses its power to destroy, while retaining its ability to teach us who we are.